Hello, everyone. My name is Asher Lull, and I'm a senior at Pelham Memorial High School. Welcome to our first ever science research virtual event. This is a collaboration between Meredith and Pelham, and we're really excited to partake in this opportunity. We thought it was important to be able to communicate and present to each other in this environment during this really strange time. Please bear with us as we iron out any kinks. We're hoping to have more of these events in the future. We will be accepting questions in between presentations. Students, please state your name, grade, and school. Everyone else, if you could state your name, institution, or field before your question so we know where your professional background is, that would be great. Finally, if everyone that just entered could mute their mic so we don't get feedback during presentations, that would also be really appreciated. Our first presenter of the night is Bernadette Russo. I will hand over the floor to her.
Hello, everyone. My name is Bernadette Russo. Let me just get my screen. Okay, great. All right. Hello, everyone. Today, I will be presenting my research that I conducted with my teammate, Morgan McLean. My research is titled Freshwater River Eutrophication, a profile of nutrient loading along the upper Hutchinson River. Nutrient loading in rivers, lakes, and estuaries has become an ecological problem worldwide. Here in the United States, harmful algal blooms from nutrient loading has caused up to $82 million in economic damage per year. The most severe economic impacts of these blooms are found in the Northeast. However, New York State has no numeric regulations on nutrient loading in bodies of water that are considered impaired. While there are guidelines and accepted levels set by organizations like the Environmental Protection Agency and the Department of Environmental Conservation, these are simply qualitative guidelines rather than implemented laws. This lack of concrete guidelines is troubling because we live near one of the most impaired bodies of water in America, the Long Island Sound. Over the years, while other portions of the Long Island Sound have improved in health, the western section of the Long Island Sound, as highlighted in red, has remained at an F grade and has shown little to no improvement. In fact, just 2 summers ago, the Long Island Sound experienced an extreme form of harmful algal bloom called rust tide. This tide shut down the beaches, harmed the surrounding ecosystem, and caused massive and unprecedented fish kills. Even though there is evident impairment in the Long Island Sound, there has been no concrete sources of this nutrient loading. However, previous research has said that rivers can deliver nutrients into larger bodies of water. Given this literature, 2 years ago, Morgan and I profiled one point along the Hutchinson, Mamaroneck, and Byron rivers, the 3 main and largest river tributaries that empty into the western Long Island Sound. We profiled these rivers for their nutrient levels to see if they were high enough that they could be contributing to the adverse environmental effects found in the Sound. We found that the Hutchinson River had the highest levels but were the most dangerous out of the other 2 rivers combined. However, the full length of the Hutchinson River has been completely unassessed on a formal scale. So that's why the purpose of our study was to profile the Hutchinson River for its nutrient levels to see if it has levels that are high enough that could be contributing to the impairment found in the Long Island Sound. We profiled 6 points along the river for their phosphate and nitrate levels, which, when in excess, introduce impairment. The average phosphate levels along the 6 points that we tested were all above the EPA healthy maximum of 0.1 part per million, and the average nitrate levels were all above the EPA maximum of 1 part per million. These elevated levels can make us conclude that the Hutchinson River is impaired based off of its phosphate and nitrate levels, showing that this river is in need of remediation and regulations. Our study is significant because it's one of the first to make a connection between the heavily impaired western Long Island Sound and the Hutchinson River in hopes of remediating one of the most impaired bodies of water in America. The study also brings the attention to the health of these smaller rivers and shows that regulations are needed for not only the Hutchinson River, but all other smaller rivers across America that empty into larger bodies of water. We are all surrounded by impaired rivers, and while we focus on the larger ones, we fail to focus on the smaller ones. This study brings light the assessment of the health of smaller rivers, because if nothing is done to stop this, then problems such as nutrient loading and harmful algal blooms will persist and cause damage to the surrounding ecosystem. I would like to give a huge shout out to my research partner of 3 years, Morgan McLean. This research was conducted last year, and Morgan has been presenting our research this year that we did on microplastic pollution. I also want to acknowledge my mentor, Dr. Thomas Callahan, for all his help and guidance, my science research director, Stephen Baltakis, my science research teacher, Joseph DeBello, and my family and friends. These are my references, and that concludes my presentation. Thank you for listening.
All right, now if anyone has any questions, just unmute your mic and then you can just spit it out, or you can type it into the chat if that's what you prefer. In the meantime, Bernadette, why are algal blooms so harmful for the environment?
So the question was, why are algal blooms so harmful to the environment? So when nutrients in excess are entered into bodies of water through anything from rain and runoff to sewage runoff, algal blooms form in excess on the surface of water. When these algal blooms decompose, they take up dissolved oxygen in the process, which deprives organisms of what they need to thrive in their environment, which further deteriorates the ecosystem.
All right, thank you. Does anyone else have any questions? I saw Morgan's comment in the chat.
Did you find that, Morgan, this is Dr. Callahan, and Bernadette, sorry, did you find that any parts of the Hutchinson River were more heavily nutrient loaded than others? And if you did, what were your reasons for those findings?
So the question was, did we find that there were any points along the river that had more elevated nutrient levels than the other points? We found that the most northern and southern portions of the river experienced elevated nutrient levels. To start with the most northern point, which was the source of the river, had very high nutrient levels, which we can speculate is because this part of the river was surrounded by residential areas. So people fertilize their lawns, and when it rains, these fertilizers contain phosphorus and nitrogen, which are the nutrients we tested, which then run off into the river and cause nutrient loading. And then the southernmost point is right next to a sewage treatment plant. So when the sewage enters, that usually contains nitrogen, so it enters that river and causes excessive nutrient loading.
Are there any other questions? If not, we're going to move on to a brief word from Mr. Garbarino, the science research teacher from Merrimack.
Thank you. My name is Guido Garbarino. I'm the science research teacher at Merrimack High School. Good evening, everybody, and thanks for joining us here tonight. I know this is a tough time for everybody, and it's really nice to see you all here together, coming together as a science research community. On behalf of my colleague Alejandro Victoria and I just wanted to thank Mr. Beltakis and Dr. Callahan and Mr. Sorry, I forgot Joe's last name, the Science Research Department at Pelham for including us in this event. And this is really exciting, and I'm just really glad that everybody had a chance to come together and present this evening. Without further ado, I'd like to give this back to Asher. I hope everybody enjoys the presentations tonight, and thank you.
All right, thank you, Mr. Garbarino. Our next presentation is Samantha Shade from Merrimack.
All right, thanks. All right. Can you see it? Okay. Hi, my name is Sam, and my project is examining the role of CD8+, CD4+, and NK cells in atherosclerosis development. So atherosclerosis is a coronary artery disease in which fats, lipids, and other materials accumulate in the arterial wall, obstructing oxygenated blood flow, and this can cause a heart attack or stroke. And that's why atherosclerosis contributes to 1 in 4 American deaths annually. So the stages of development have been elucidated. First is lesion development. These are just tears in the endothelial lining that give the plaque a position to accumulate. Next, the body recognizes that there's something wrong, and this triggers the innate immune response, which contains NK cells. And then this triggers the adaptive immune response, which contains CD8 and CD4+ T cells. So again, these 3 target immune cells in this project are the NK, CD8, and CD4+ T cells. So there are 2 main functions of the NK cell. On the right, we see the secretion of cytotoxic granules. This leads to premature cell death in a target cell. And on the right, the secretion of cytokines, which just affects cellular signaling. Next, CD4+ T cells they secrete cytokines, which is this molecule that's released and interferes with cellular signaling. And CD8+ T cells is the secretion of cytotoxic granules, which directly enter the cell membrane and lead to apoptosis. So next, we know where these cells reside under normal conditions. So the NK cells are in the lymphatic tissues, which are highlighted in yellow, And the CD4 and CD8 are also in these lymphatic tissues. The only difference is they mature primarily in the thymus. The most important thing to note is that none of these immune cells are in the aorta under normal conditions. So this leads me to my question: do T cells and NK cells of the immune system, do they migrate to the diseased aorta from the lymphatic tissues where they normally reside? And is this associated with atherosclerosis development? So next, the methods we're using are two mouse models, the B6 and the ApoE knockout. Both are susceptible to high-fat diet to atherosclerosis, and they were separated into two diet groups. So first, the chow is the control diet, and high-fat diet is the American Western diet here. And then 6 tissues were taken here and put through a flow cytometer. And this is like a funneling effect. So you start with a tissue and you try to isolate specific cell populations. So in step 5, we see the isolation of the NK cells, and in step 6, the isolation of CD8 and CD4 plus T cells. So here we see each bar represents a different mouse, and this shows the percentage of CD4 plus T cells out of all immune cells in the spleen. And then we created an average between the chow and the high-fat diet just to show the percent change from healthy to diseased. So moving on to the results for the NK cells, there's an increase in the aorta. So when the disease is present, the NK cells are entering the disease plaque, and there's a decrease in the spleen and lower lymph nodes and upper lymph nodes. So there is this general migration pattern out of the lymphatic tissues into the aorta. For the CD8 plus T cells, there's an increase in the aorta again, and a decrease in most of the lymphatic tissues. So like the NK cells, the CD8 plus T cells exhibit a migration pattern from tissues they're normally in to the diseased aorta, with the exception of the liver. And finally, the CD4 plus T cells, there was a decrease in the aorta and all the other lymphatic tissues. This was different from the other 2 immune cells, and it does not show the same migration pattern. So again, there was an increase in the aorta of the NK and CD8 plus T cells under disease conditions, and a general decrease in the lymphatic tissues, showing this migration pattern. Comparing this to the hypothesis, the CD4 plus T cells did not exhibit a migration pattern that the NK and CD8 did, and the thymus and liver were not really predictable. So the main thing to take away from this is that the NK and CD8 plus T cells did exhibit this migration pattern out of lymphatic tissues into the diseased aorta, while the CD4 plus T cells did not. And this is significant because it indicates that disease presence disrupts normal mechanisms of these immune cell migration patterns. And because the NK and CD8 plus T cells are entering the plaque during disease presence, it can be said that these cells contribute to the cytotoxic environment surrounding the plaque, and this can be a target for future treatments. These are my references, and thank you for listening. Do you have any questions?
All right, thank you, Samantha. Does anyone in the audience have a question? Okay, well, is the immune response considered helpful or harmful when heart disease is present?
So generally with atherosclerosis, normal body responses like the immune response and inflammatory response actually increase the size of the plaque and create this cytotoxic environment in the aorta. So suppressing these responses can actually slow progression, and this is becoming a target for future treatments.
All right, thank you. Anyone else? Okay, I guess not. So let's move on to our next presenter, Olivia Min from Pelham Memorial High School.
Hi, my name is Olivia Min, and today I'll be presenting to you on the registration accuracy of augmented reality for neurosurgical navigation. Currently in the United States, there are about 700,000 people with brain tumors, and it's estimated that in 2019, about 86,970 people were diagnosed with primary brain tumors. Because the survival rate for people with brain tumors is about 35%, those who are diagnosed don't often survive for more than 5 years after their diagnosis. However, in those that do survive, brain tumors are known to lead to lasting cognitive, psychological, and physical effects. Currently, to remove a brain tumor with surgery, this requires one to first target the tumor, and many of these current targeting strategies rely on image-guided neurosurgery, or IGNS, which uses preoperative images of a patient's brain in order to find the best way to get to the tumor. However, in past research, it's shown that procedures that rely on IGNS are known to have workflow disruptions because neurosurgeons must look up from the operating field to view images of the patient's brain, which leads to these disruptions. Therefore, it's necessary to look for new ways to target brain tumors, which are both more time efficient and effective. So one way that we can potentially target brain tumors while avoiding these workflow disruptions is by using augmented reality. In past research, both augmented reality and virtual reality have been used to project images of the brain and also to view the anatomy of the brain. However, many of these studies have been hypothetical or completed using virtual diagrams and references for projections, such as a study completed by Mavash and Tabrizi in 2013. Additionally, studies that have specifically used augmented reality to target tumors in the brain, many have done so in ways that are not feasible for actual use, such as a study conducted by Hugh and others in 2016, who used a smartphone to target a tumor in the brain. Also, there's been no previous research using the Magic Leap AR goggles specifically to target brain tumors, so in my research, I was looking to do this. Prior to brain tumor removal procedures, neurosurgeons must often rely on functional magnetic resonance imaging in order to avoid removing critical structures in the brain. This involves the overlapping of many different images, and so it can therefore lead to a mismatched final brain image. So to combat this, neurosurgeons employ linear MRI image registration, which is the geometric alignment of one image onto another, and this creates an usable image that neurosurgeons can now rely on to perform a brain tumor removal procedure successfully. However, in the past, it's been debated whether manual or automatic registration is better. While some argue that manual registration is better, others say that automated registration is faster and more easily reproduced and will therefore be more beneficial to neurosurgeons in the future. However, it's still unknown which software for performing automated registration is the best. So in my research, I was looking to assess two softwares, which were 3D Slicer and FSL, in order to see which one was better for performing image registration. After assessing the accuracy of the AR projection for identifying a tumor in a 3D printed model of a patient's brain, we found that it had a registration error of about 1 millimeter, which means that it was about 1 millimeter off when projected over the 3D print. Concerning the image registration aspect of my project, we performed 4 different types of registrations using each of the programs for each of the 10 subjects, and this included mapping DTI, T2, T2 FLAIR, and SWI images onto T1 images individually. And then after comparing the accuracy of both programs, we found that FSL was more accurate for performing image registration. However, many companies today still use software that's like 3D Slicer software, primarily because it's easier to use and it also takes less time, although as we found in this study, it is less accurate. So in the United States, there are currently about 700,000 people with brain tumors, and it's estimated that in 2019, about 86,970 people were diagnosed with primary brain tumors. In the past, where IGNS has been used to target tumors before brain tumor removal procedures, this has been known to lead to workflow disruptions. So to avoid these workflow disruptions, we can potentially use augmented reality to target the tumors instead. However, many past studies that have done this have done so in ways that are not feasible for actual use, but using the Magic Leap AR goggles can easily be implemented into neurosurgeons' brain tumor removal procedures, therefore making the procedure easier to perform, leading to shorter procedural times, and ultimately lessening the risk of brain infection. By using the Magic Leap AR goggles to target and remove tumors, we'll eventually be able to target and remove entire tumors accurately and efficiently, therefore leading to an overall increased patient quality of life. I'd like to acknowledge my mentor, Shumit Yogi, Sneha Pandya, who was a research data specialist in the lab who helped me, James Shin, my science research director and teacher, Stephen Baltakis, and then also my family. These are my references, and that concludes my presentation. Thank you.
Thank you, Olivia. Does anyone have any questions at all? How do you make the AR projections?
Okay, so to make the AR projection, we first perform tractography on a patient's brain who we were going to use to make the AR projection. And what this does, it allows you to generate the nerve tracks running through the patient's brain, which can't be removed during the procedure. And then we also generated models of different important structures that would be necessary to see when you're performing the procedure, such as models of the tumor, the ventricular system, the vasculature system, and a couple other models. And then we put them together so that they can be accessed from the interface of the Magic Leap AR goggles and then projected over the 3D print or the head.
All right, thank you. Does anyone else have any other questions from the audience?
Olivia, I'll ask a quick question. What's the barrier of entry for this? Like, are these goggles really expensive or no?
No, they're not really expensive, but they're fairly new, so there's not been like a lot of research using them, but I think they could be easily implemented with practice, because we did find that there was an error of about 1 millimeter, which indicates that it's not yet ready, because ideally we want to have no error before we use it in an actual procedure, but it's on its way to getting there.
All right, thank you, Debella. Does anyone else have a question? No? Okay. Oh, in the chat, Olivia, do you want to respond to that? What level is acceptable?
Okay, so in terms of performing like a brain tumor removal procedure, ideally you'd want there to be no error, because there always would be maybe a small error. So you wouldn't want there to be any error that you can detect, because otherwise that could add up and mess up the procedure. So the acceptable level of error would be no error, ideally.
I have a question.
Go
What
ahead.
about lag time? Because it would take a few moments for the screen to refresh and for the signals to be sent. So what about lag time with the goggles?
Oh, well, we didn't necessarily look at that in my project because we're just looking at a 3D printed model, but I think that's definitely something in the future that we should look at, like when we're actually using it on a person and testing it like that. So that's a good idea.
Any other questions?
I have a question. I'm Spencer Wolf from Mamaroneck. In the future, could these goggles somehow like be shrunken and be more widely distributed?
Yeah, I think that would be a really good idea, and especially if they're made like slightly more affordable so that a lot of different institutions can have them. I think that would be a really good idea.
Interesting. Thank you for answering my question.
All right, thank you, Spencer. Does anyone else have any questions before we move on? All right, and before our next presentation, a word from Mr. Beltakis, the Science Research Director at Pelham Memorial High School.
Hello, good evening, everybody. Thank you very much for joining us tonight. Mr. DeBello and I and Mr. Garbarino are very excited to be hosting this event for everybody. Great series of questions that just came at us. Please, we are encouraging questions. So at any point, whether you want to throw them into the comments or you want to just jump in live, people that are watching us on YouTube right now, we apologize. We don't have that feature set up for this time around, but hopefully on the next round, we will be able to set that up for everybody. So in terms of what we're doing here tonight, we wanted to give everybody an opportunity to be able to, as we're locked kind of at home and practicing social distancing, we really wanted to showcase the work that our students have been doing with science research over the year. And especially our seniors, as this is their last little push here in the last couple months, we wanted to give them another authentic avenue so that they could get out there and present their work and field questions from the audience. So all of you joining us again, thank you very much. Thank you, Mr. Ravita, who actually is out there setting up our live stream with YouTube. We appreciate your help. And a special thank you to Dr. Callahan, who's always supportive of pretty much all of our teachers as we look to try to bring something next level for our kids. So Dr. Callahan, thank you in terms of getting us, helping us to get this off the ground. We're really excited about this opportunity. People that are out there from the community, whether you are from Pelham or whether you are from Mamaroneck, please reach out to us. As we go into the summertime, students are going to be looking for opportunities that might be, they're not going to be able to get into a lab situation, and it's going to be very difficult for our students to be able to work in the situation based on the climate of what's happening in the world. So we are looking for some type of remote mentor system where maybe 2-3 times a week you're able to remotely work with kids in this format, and then the kids can kind of feed their ideas and you could bounce them off you. So please, if there's anyone out there that can be helping our students or you know somebody that might be interested, have them email either Mr. Garbarino over at Mamaroneck or myself or Mr. DeBello over in Pelham. Thank you all again, presenters so far. Great job, Asher. Thank you for moderating tonight. And please keep those questions coming. Our students love when the questions are asked. And as you can see, they're ready for it. They've been working for this for a long time. Thank you.
Thank you, Baltakis. Our next presenter is Matthias Spunberg from Pelham Memorial High School.
Amazing. There we go. It's 4 a.m. You have a presentation tomorrow, but you're barely halfway through finishing it. You wonder where the time went, how you let yourself do this, why you procrastinated so much. Good evening, ladies and gentlemen. My name is Matthias Spunberg, and I'd like to discuss my work with procrastination. It's from this kind of situation that I set out to understand public needs regarding a potential new method of managing needless delay. Ellison now has found that 95% of American adults are prone to procrastination in their daily lives. While this number is no huge surprise, it's also estimated that as many as 1 in 5 adults are chronic procrastinators, whose behavior can be self-destructive to various degrees of seriousness. Many people may know that procrastination is stressful. In addition, however, procrastinating lowers academic performance, has negative impacts on mental health, and causes greater exacerbation of illness. Furthermore, Nguyen and Steele estimate that employers lose nearly $10,000 per employee per year through lost productivity, amounting to a national total of $85 billion in a single year. People often assume that procrastinators have laziness or inability to manage their time. As such, time management training would seem to be the best way to minimize procrastination. This idea is reflected through various sources, including researchers, self-help authors, and productivity or time management apps. This idea is widespread, but is actually incorrect at its core. Procrastination is not just laziness that can be solved with a planner. Rather, a multitude of studies have shown that unregulated negative emotion is actually a primary cause of procrastination. This unregulated negative emotion can have the following impact on one's work. When presented with the task, a person always faces 2 options: do the task or avoid it. Doing the task is productive, and avoiding it is procrastination. Every time the task is avoided, they face the same question. This continues on until the task is completed, or the task can no longer be completed, typically due to overdue deadlines. When faced with unregulated negative emotions, a person can favor seeking a short-term mood booster through distraction, rather than long-term satisfaction through work. Therefore, task avoidance becomes more likely, which blurs the path to timely task completion. By providing an intervention that tackles emotion regulation with a training program, procrastinators can decrease the prevalence of procrastination in their lives by addressing one of the biggest factors causing this procrastination. Existing productivity management solutions cannot cater to procrastinators. In order to build a more effective one, though, user preferences for medium and methods used in the new procrastination management intervention are necessary. Thus, the purpose of my studies is as follows. I set out to establish an informational base of knowledge for the future development of a procrastination management intervention, or PMI, by first identifying a need for a new PMI, and then identifying preferred management methods and media that can combine with emotional regulation for the creation of an effective PMI. I conducted my research over about a year and a half using 6 surveys that gathered 6,388 participants. I split into 3 main phases, the first being an exploratory phase in which I created a base of knowledge that would help guide future survey development. Here, with Survey 1A, I looked to identify the favored management methods from a greater list from multiple sources and found these 6 methods to be favored among my participants. After this, I moved on to Survey 1B, where I looked to identify openness to new PMIs. I found that participants were open to new PMIs across age groups and genders, as well as a number of other demographics, although less strongly. After this, I moved on to Phase 2, where I created and distributed a validated survey that assessed needs for a potential new PMI. I gave participants 7 statements and had them identify their agreements with the statements on a Likert scale. We see that the averages, ranging from 2.7 to 4.2, lie in the positive range of the Likert scale, suggesting that there is in fact a need for a new management intervention. After this, I moved on to Phase 3, where I created a survey that looked at method and medium preferences for new PMI. I had participants identify their top methods and found that large task breakdown and task prioritization, with more than 2/3 of participants, were the most popular methods. After this, I looked at media and found that mobile applications were the favored method, with more than 37% indicating that this was their favorite method for an intervention. So I intend to apply this work in the future, as I will create an app that leverages preferences for methods such as task prioritization, schedule creation, and the Pomodoro method, among other things. More importantly, I'm going to incorporate the emotion regulation techniques from cognitive behavioral therapy in order to allow my participants to self-regulate their negative emotions, potentially providing millions with an accessible, effective approach to managing the primary cause of their procrastination. This intervention can provide the 95% of adults who procrastinate on a regular basis with a way to lower their stress. This intervention can help the 20% of chronically procrastinating adults to take control of their detrimental behavior. Most importantly, this intervention addresses a fundamental human flaw, the irrational behavior that we've all been guilty of partaking in before. Procrastination does not have to cost billions or stress out millions of Americans, because moving forwards, there's now a clear path to an accessible, effective solution. I'd like to thank my mentors, Dr. Pierce Steele from the University of Calgary, Dr. Thomas Callahan from Pelham Memorial High School, as well as my research teacher, Mr. Joseph DeBello, and my research director, Mr. Stephen Baltakis. These are my references and more, and that concludes my presentation.
All right, thank you, Matthias. Does anyone have a question?
I have a question. My name is Sid. So my question is, during your study, what was the biggest hurdle you faced from the surveys you were taking out?
Right. So the question was, what was the biggest hurdle I had faced? Moving from Survey 1A to Survey 1B, I had an issue with sample size. Survey 1A, I managed to get 37 participants, which was not a very big number, but it was good enough for a pilot. However, I knew I needed to make some changes to my survey, so I looked to shorten questions, shorten the amount of questions, make the wording easier to understand, and change the method of advertising, which allowed me to increase my sample size from 37 in Survey 1A to 3,607 in Survey 1B. And so after having spent a lot of time making those changes, I was able to apply that to future surveys, and that was a lot easier from then on.
Great, thank you.
All right, thank you, Mr. Jane. Does anyone else have a question? How did you manage to get over 6,000 participants? Piers, you can ask afterwards.
Okay.
All right. After this, I sort of answered that. Basically, it was a matter of shortening the questions, shortening the amount of questions, and making the readability of each question brought down to a more standardized lower level so anyone can understand it regardless of education or age.
All right, thank you. Piers, do you want to ask your question?
Do you think that this would have a greater effect on people with mental illness such as depression or ADHD, since you say that it mainly that you're trying to focus on negative emotional suppression, that sort of thing?
Right. There has been a large amount of research done on links between procrastination and depression and anxiety in particular. It was previously thought that anxiety is that procrastination was split into three main types. There were people who avoided procrastination for the thrill of it, people who avoided it because they were perfectionists and didn't know where to start, and people who avoided it because they were too anxious. Now that's been disproved, but the link between anxiety or other mental disorders and mental health issues like that still is very strong and does exist. Because as you say, these do bring about more severe negative emotions that are more difficult to control.
All right, thank you. Does anyone else have a question? If you don't feel comfortable saying it out loud, you can always send it in the chat.
What sets your proposed app apart from other apps on the App Store?
Right, so the question was, what sets my app apart from other apps in the App Store? So the apps that we currently see available to the public, what they are is not targeted procrastination apps, but rather they're productivity apps. So what they feature are productivity techniques such as large task breakdown, the Pomodoro method, and things like that, but they do not feature that crucial emotion regulation element that makes it targeted to procrastination. So by introducing this emotion regulation aspect, what you can do is make people more likely to use those productivity methods to the best of their ability. And so that will make it more effective and more available to a wide range of procrastinators.
All right, thank you, Lucas, for that question. Is there anyone else before we move on to our next presenter? Okay, our next presentation will be from Ravi Bala from Mamaroneck High School.
Hi, everyone. I'm Ravi Bala. Let's see. Okay. Okay. So hi, everyone. I'm Ravi Bala. I'm a senior at Mamaroneck High School. I'll be presenting my work on 2 novel approaches to optimize the SRC kinase biosensor using fluorescent protein insertions. Now to begin, most have heard of a disease known as cancer. It affects millions every year and costs us hundreds of thousands of lives. Because of this novel significance, it has driven a large portion of research in recent history. While we have made some effective therapy strides, we have yet to unlock a single cure for such a complex genetic mutation. And the key to this is by redefining our approach. Now, when we speak of redefining our approach, we look at what are known as key downstream indicators. And the goal of this is to identify the root causes of cancer and inhibit them before they actually mature. One key root cause studied by Wheeler in 2009 is the SRC kinase enzyme. It controls some pretty basic functions in the cell. However, it has been shown to have a novel role in the spread of cancer. And to learn more about the specificities of its activation, we look at what are known as, we do this through biosensor technology. And biosensor technology is a well-known and well-studied method in molecular biology that focuses on using one biological molecule to detect for the presence of another in a cell. And in this study, we aim to design 2 effective designs to this biosensor. Our first approach was to use tension-sensing fluorescent proteins and insert them into the SRC kinase. And the second approach, which will be examined independently, was to substitute these proteins with what are known as FRET proteins. To begin, we have a figure here on C-SRC activation. As you can see, SRC kinase switches between an inactive and active conformation. And the goal of our first design was to insert some tension-sensing fluorescent proteins in order to capture these conformational changes with fluorescence modulations. Now, to start, you can see the 2 proteins here. We inserted a green fluorescent protein in an active linker in between the SH3 and the SH2 domains. This was to provide modulated fluorescence. And we secondly inserted MIRFP703. It's a red fluorescent protein. It was inserted at the very end of the complex at a very inactive linker in order to provide unmodulated fluorescence. And the goal was to basically divide these 2 and notice our fluorescence modulations overall. Our main method here was to insert a restriction site known as Hindi3. This allows us to cut and paste a little more efficiently. And we were looking to substitute the sequence SH3 and the kinase with mutants in order to quantify our results on a deeper level. Following that, we used overlapping PCR. This is a cycle of PCR that uses 2 cycles. And we followed that up with ligation, which uses mutants and vectors and mixes them with a ligation enzyme. We then prepared the entire complex in bacteria with an SOC medium, a carbonicillin antibiotic. Then we sugared overnight. We then picked colonies the next day and processed it for DNA purification, which was later confirmed by sequencing. And then we transfected this all into HEK 239 T cells, which are human embryonic kidney cells, to image and process them. For our first preliminary results, we looked at the following images. As you can see here, the key aspects to notice are the bright fluorescence patches, which indicate adequate protein folding. When we moved to quantify our results on a deeper level, we looked at fluorometry with 4 groups and 2 wild-type samples, starved and serum stimulated. The serum stimulation was intended to induce an activation that would produce the conformational changes and thus the fluorescence modulations that we hoped. And as you can see here, there's a key discrepancy between the 2, which can be quantified as a 64% increase, which indicates that the cells in the biosensor were adequately responding to the stimulation we introduced. As we looked at our second approach, we substituted these proteins with FRET substitution. So FRET is Forster resonance energy transfer. It uses 2 fluorescent proteins to detect for the opening and closing of a certain molecule. The 2 that we looked at were a donor and acceptor. This is a known FRET pairing that uses M cerulean 3 and M venus to substitute for the 2 proteins we looked at before. Our methods were largely the same, however, we used a different fluorometer. This is the Fluorolog 3 spectrofluorometer from Hariba, which measures fluorescence emission on a deeper level using a multi-well plate reader to detect more efficiently for FRET. As you can see here, when we quantified our results a little more, we looked at the conformational changes with wild type attached and detached, having low or having high and FRET peaks, high and low FRET peaks respectively, which can be quantified to 225%, which indicated to us that both versions adequately respond to stimulation and cellular behavior. As for our conclusions, as I mentioned before, our data plot showed a response to overall stimulation with very significant change visualized. These were positive initial findings. However, for our limits, due to the nature of this trial being very trial and error, the results are hard to replicate or predict and contain very high degrees of variability. As for our future, we want to move a little bit past these preliminary successes, focus on testing the E and F mutants mentioned before to refine the effectiveness of the sensor a little bit more. We're next going to try different FRET pairings that have been studied in depth before that might have the potential to be a little more efficient. Primarily, this identifies very preliminary results in a novel methodology. However, on a broad scheme, it shows that this is a very new idea and our approach to cancer research needs to be redefined. We need to identify the root causes of cancer and find new ways to inhibit them, which means that we're contributing to trailblazing a new path for the most important part of modern research. I would like to thank my mentor, my parents, and my research teacher for support throughout this whole process. My work would not have been possible without them. My references are here if we have any questions, and refer to specific parts of this slide. But as for now, that was my presentation. Thank you for listening.
All right, thank you, Robbie. Are there any questions on the floor?
I do have a quick question for Robbie. I'm Peter. I'm from Pelham High School. So I was wondering, so the enzyme you're studying, right? What kind of cancers has it been implicated in to be like the root cause of?
Yeah, so as of right now, we have novel significance that have been studied and tied with melanoma, ovarian, and breast cancers. However, we anticipate that if we study it a little more in depth, we're going to refine what exactly it controls to a deeper extent, maybe find some more types of cancers that it actually controls, and maybe, refine the effectiveness a little bit more.
Thank you.
All right, thank you, Peter. Is there anyone else with a question?
I remember seeing yours at JSHS, and I was wanting to ask then, this idea of using a glowing protein on cells, do you think it could be applied on a larger scale for not only cancer, but sorry, I'm forgotten my question.
That's okay.
I'll leave it to the floor for a moment while
Okay, does anyone else have a question in the meantime? In the first approach with attention-sensing fluorescent proteins, was there a specific reasoning behind the exact fluorescent proteins you chose in their placement?
Yeah. Okay. So essentially, we used for the green fluorescent protein that I mentioned before, we used what is known as CP145-MEGFP, and this was chosen very specifically as its structure is made up of a beta sheet barrel, which inside contains a chromophore. And essentially, this beta sheet barrel acts almost like a Chinese finger trap with the conformational changes, placing it in active linker like we did. This beta sheet is supposed to constrict or relax to modulate the fluorescence that is emitted from the chromophore within. As for our second protein, which was MIRFP703, we essentially just chose it as a standard red fluorescent protein. And what was significant mostly about that was its placement. We put it at the very end of the enzyme and the complex, as it's an inactive linker with not a lot going on.
All right, thank you. Piers, you collector. Okay.
So what I was really wanting to ask is, how invasive is this? Because you're making something glow. Is the person going to have a glowing patch on their arm or something? Or what are you really doing? And I know that you said this was unpredictable. What are you going to do to make it so that we can identify the glowing?
Okay, yeah, so that's a good question. The question was what we would do to inhibit basically like the glowing in its application, I guess. So basically, the goal of this study in particular was to use these fluorescent molecules to detect for the presence in a certain biosensor. So we use them in an adaptation in order to basically identify SAR kinase in cells, quantify its concentration, and the specificities of its activation. So essentially, the fluorescent proteins are basically used just to refine the effectiveness of it. And then once we target basically how the SAR kinase controls cancer metastasis on a very specific level, we'll then look into more advanced therapy options. So the fluorescent proteins are used more in studying SAR kinase a little more in depth. And as for cancer therapy, we would be looking towards another field for that.
All right.
Thank you.
And Robbie. Does anyone else have any other questions before we move on to our next presenter? No? Okay. Our next presenter is Diana Haggis from Meredith High School.
Awesome. Thanks. All right. Cool. All right. Like Asher said, my name's Liana Haggis. I'm from Meredith High School, and my study focused also on cancer treatment like Ravi's, but I went at it from a bit of a different approach in an immune system-based therapy. So my project was HIV-1 GP41 3S induced surface NKp44L expression as a novel target for pancreatic cancer therapy. So before tumors can grow into malignant cancers, they have to first evade patients' immune systems. And there are many different ways that cancers do this, but one important one is called the loss of antigenicity. So you can see here in this cell that the tumor cells have lost antigens that activate an effective immune response in the patient. So this makes it difficult for the patient's immune system to identify the tumor cells as cells that should be disposed of. Now, luckily, there is a protein called HIV-1 GP41 3S that can be used to identify these malignant cells. So in HIV, this protein GP41 is first secreted by infected CD4 positive T cells, which then bind to a cell receptor known as GC1QR on other T cells, which activates a complex signaling cascade within the cell that results in the translocation of a natural cytotoxicity ligand known as NKp44L to the surface of the cell, and therefore initiates NK cell mediated apoptosis. So the key to this is that GP41 and its interaction with this T cell in HIV identifies cells for destruction by patient's immune system. So, because GP41 has this effect in T cells, I wondered why not cancerous cells, to be able to identify those as cells to be destroyed by patients' own immune systems. So my hypothesis was that cancer cells, which express the same cell receptor that T cells do, GC1QR, would, after GP41 exposure, expose NKp44L on the surface of the cells and therefore be identified to the patient's immune systems. Moving into my methodology, I first cultured the pancreatic cancer cell line, PANC1, according to laboratory protocol, and sought to confirm that GC1QR was in fact present on the surface of these cancer cells, and so GP41 would be able to potentially bind to them. Next, I completed an ELISA. Well, to confirm this presence of surface GC1QR, I completed a whole cell ELISA and confirmed that this receptor was in fact present on the cancer cells. So then I moved into my next phase, which included exposure to GP41 and the specific protein motif GP41-3S that binds to GC1QR to see if it would have the same effect on the cancer cells as it did in T cells. So for this, I first cultured my pancreatic cancer cells with GP41 or that specific 3S motif for 12 hours and completed another ELISA to test for surface NKp44L to see if it had translocated from the inside of the cancer cell to the outside, as I was expecting. So after 12 hours of GP41 incubation, I saw no statistically significant increase in the level of surface NKp44L. So I adjusted my incubation time to see if changing that variable would produce the outcomes that I expected. So next, I completed the same procedures as the last experiment, but exposed the pancreatic cancer cells to GP41 and GP41-3S for 72 hours instead of 12. Now, after this, I saw many statistically significant increases in the presence of NKp44L, which supports my hypothesis. So this graph shows GP41 concentration and its effect on surface NKp44L expression after incubation. So there were statistically significant increases at 5, 50, and 100 micrograms per milliliter, with a direct relationship seen between those values. So again, this proves that GP41 did have that same effect as it does on T cells on cancerous cells, therefore identifying the cancerous cells. This graph shows GP41-3S, or that specific motif, and its relationship between concentration and surface NKp44L expression, which again shows that exposure to this protein did identify those cancer cells. Now, between the two, the GP41-3S exposures did create higher translocation of NKp44L to the surface of the cell as compared to GP41, the whole protein. So in my first phase of the study, I confirmed that GC1QR was present on the cancer cells, then exposed them to GP41, and found that after 12 hours, there were no statistically significant increases in NKp44L, but after 72 hours, there absolutely were, which supports my hypothesis and shows that GP41 can be considered as a new cancer therapy. Now, again, all my results supported my hypothesis. And in the future, I seek to co-culture treated cancer cells with NK cells to see if that reaction of the NK cells destroying the marked cancer cells does indeed happen, which would be very exciting. So my study was the first step in developing a new immune system-based cancer therapy that could hopefully provide relief to the millions of patients who are diagnosed with cancer every year. I'd like to thank my research teacher, Mr. Garbarino, my colleagues at the lab, my research mentor at Stony Brook University, and my family for all their support. Here are my references, and I'd be happy to answer any questions you have. Thanks so much.
All right, thank you. Are there any questions from the audience?
Hi, I have a question. I know you did a study increase from 12 hours to 72 hours. Did you think of any other variable, like going 144 hours or a different variation?
Yeah, so that's actually a common question I get. Why didn't I do a gradient, or why not more or less? At that point, I did my research over this past summer at a lab on Long Island, and at the point where I was able to do that phase of the research, I had to make the best of the remaining lab time that I had. So I just went from 12 to 72 to see if I would get results that supported my hypothesis at all. And then I knew that if I found that in the future, I would be able to test times in between to find that optimal incubation time, or even incubation time longer than that to see if that provides optimal results. Thanks.
Thank you. That makes sense.
All right, thank you, Mr. Jain. Is there anyone else with a question?
Yeah, I have a question. Hi. I was just wondering how would you prevent your treatment from targeting other cells that also have the same receptor that you were looking at and preventing preventing those cells from dying along with the cancer cells?
Yeah, so that's going to be a huge consideration going forward with this project, because GC1QR, that receptor that GP41 binds to, is present on most cells in the body. So of course that would be problematic just sending in GP41 molecules willy-nilly. So through some biomedical engineering work, which we've already started proposing ideas in the lab, we're going to try making a drug delivery mechanism that only allows the GP41 to bind to cancer cells expressing certain other receptors that are specific just to cancerous cells to be able to more specifically target. But that's absolutely a huge concern that we're absolutely addressing going forward.
Thank you.
Thanks.
All right, thank you, Peter. Is there anyone else with a question before we move on to our next presenter? No, all right, thank you so much. Our next presenter will be Catherine Tubner from Pelham High School.
Good evening, my name is Catherine Tobner, and I'll be presenting on using annotations in combination with machine learning as a novel approach for extracting medical information from imaging. So what this means is using machine learning to find medical information. Machine learning is the ability for a system to autonomously gain knowledge by picking up on patterns in large data sets. Therefore, your overall goal is to use machine learning algorithms and medical images, take information from those images. For example, a person has diabetes or their ejection fraction. This method is non-invasive and is also efficient. When a person has diabetes, their pancreas is affected in its volume, diameter, and fat content. Also, nuclear stress tests can be used to see if there's any blockages in a person's coronary arteries. The ejection fraction is a value that can be found from nuclear stress tests, and it's the amount of blood that gets pumped out of the ventricle versus the amount of blood that's in the ventricle. Previously, it was found that when using a fully automated process, it only took around 8 seconds to find the ejection fraction's rotation. Therefore, my gap in research is that this is the first time that machine learning has been used to detect diabetes mellitus using CT scans of pancreas, and SPECT images specifically were used in combination with machine learning to find the ejection fraction of the patient. So for detecting diabetes, the first step was complete annotations of CT scans of the liver and the pancreas, and then to use machine learning in order to detect if people had diabetes based on the annotations. For finding ejection fraction, the first step was also to annotate images from nuclear stress tests, and the second step was to find ejection fractions from those annotations and verify them. So for finding the diabetes, the first step was to annotate. This was done in Adobe Photoshop, and hundreds of CT scans just like this were annotated. So either the liver or the pancreas is annotated. In this case, it was the liver. And what annotating is the outlining of the organ. So once the annotating was complete, the mask could be created. Then the machine learning portion was completed. The results show the learning rate first, and the number of steps correlates to how many times the entire data set was fed into the algorithm. The learning rate differed because the data shouldn't be overfitted or underfitted. Also, this shows the entry loss, and the higher the entry loss, the lower the accuracy. Mean dice score is 0.73, and what the mean dice score is the correct overlap between the ground truth mass, which is the mass of a healthy liver pancreas, and the predicted mass. Ranges from 0 to 1, and 1 is the most ideal number. Next, for detecting the ejection fraction, the first step was complete annotations. This is a single frame, and exactly 400 frames were annotated by myself. And going to each frame, a red dot was placed on the brightest points, and because this represents where the most blood is, and the annotation was complete once around 50% gray was reached. This is when the blood isn't really as apparent. Then the ejection fraction had to be found, and to do this, the dots were connected, and then the volumes were found. Then the equation seen at the bottom of the screen is what was used to find the ejection fraction. In order to verify them, the ejection fractions from a commercial automated medical imaging software were compared to these manual annotations. ejection fractions. This represents the comparison between the manually annotated ejection fractions and the commercial software ejection fraction. It had a correlation value of 0.59 and a slope of 0.4. So the reason why CT scans can be used to find diabetes and why it's useful that CT scans can do this is because it's found that around over 70 million CT scans are received by Americans each year. This means that a lot of people receive CT scans, so they can be repurposed in order to find diabetes. Also, this process can determine health of individuals currently and in the future, and it's the first time that machine learning has been used with CT scans of pancreas to diagnose diabetes. Next, the reason why the end goal is to find the ejection fraction using machine learning is because manual annotations can take a lot of time. So for example, it took me around an hour to complete an annotation, while previous found that it only took 8 seconds for an automated process to find the ejection fraction of a single patient. Next, it also offers a way to standardize finding the ejection fraction, because depending on who's doing the annotating, the ejection fraction could differ. And the reason why ejection fraction is such an important value is because it's the only quantitative way to see if a person's heart is healthy. And overall, this method of annotating can be used to find medical information from medical images. And it's a very diverse method, as seen through how it can be used to detect diabetes and find the ejection fraction of patients. I'd like to thank my mentors, Subi Alareth from Weill Cornell Medicine, Benjamin Lee, Rupit Sai, Gabriel Maliakal, Nasu Gamal, my science research director, Ms. Beltakis, and my family. Use my references, and thank you for listening.
All right, thank you, Kath. Are there any questions on the floor right now?
Kath, can this be used for organs other than just the pancreas? Could we do this sort of scan for a heart or a stomach or various other organs, or is it purely for the pancreas?
Yeah, this could definitely be used for other organs. So for example, 2 years ago when I was doing the diabetes part, they were also using this method for the heart. So I think as long as there's a change in some shape of the organ, that could definitely be used to detect other diseases with other organs.
Okay.
All right, thank you, Piers. Any other questions?
Hi, Catherine. My question is more on the process of annotation. When you talk about machine learning, can you elaborate a little bit more on the dotted method for smaller objects than the size that you studied on?
So the question was for the annotating part, how the machine learning detected it.
Yes.
Okay, so so the images were fed into there's certain images for detecting diabetes that were set to be training and set to be testing. So around 80% of the images were used to train the algorithm, and then the 20% of the images were used to test the algorithm. So the algorithm would be trained, and once it was trained after, it would be tested, and this process was repeated until the highest accuracy rate was found.
Makes sense. Thank you.
It's Rob Min. How are you?
Good.
Great
How are you?
job.
Good.
I have a question. In addition to just identifying the pancreas on CT scans, were there particular characteristics of the pancreas that were correlated with diabetes? Because the pancreas in itself, doesn't correlate. Were there changes in the volume? Were there certain shapes that led you to correlate that with diabetes? I'm just curious. Maybe I missed it in your presentation, but it wasn't clear to me.
Gotcha. Yeah. So the diameter and the volume of a person who has diabetes of their pancreas changes. So for this project, just the shape of the pancreas was used to detect diabetes. So since the diameter and the volume both change, this would allow for the shape to be contributing to detecting diabetes.
Great. Thank you.
All right, thank you, Mr. Min. Anyone else with questions? No? Okay, let's move on to our next presenter, Caitlyn Rogoff from Meredith High School.
Hi, everyone. So my name is Caitlyn Rogoff, and I'm from Meredith High School. I'm a senior at Meredith High School, and I studied the impact of maternal anxiety on infant reaction to stress during early development.
So what many people don't know is that adolescent anxiety can be traced back to habits that were originated during infancy. And this can manifest itself in forms of academic anxiety and social anxiety. So for this specific project, I would like to define maternal anxiety. So maternal anxiety is a measure of the anxiety and stress that a mother feels after delivering her infant. In 2010, Cates used the still face paradigm to mimic a stressful situation. Infants of mothers who have a higher anxiety levels were less likely to show a negative reaction during this reenacted stressful situation in the lab because these infants see this type of behavior from the mother all the time at home. So in the lab, they weren't really caught off guard by it. And in 2014, Shea concluded that there was an inverse relationship between socioeconomic status and stress. So going into the goals of my project, I first wanted to determine the relationship between maternal anxiety and an infant's reaction to stress. I then wanted to determine the relationship between maternal anxiety and socioeconomic status. I first hypothesized that higher levels of maternal anxiety will be correlated with infants displaying less of a negative reaction to stress. And second, lower socioeconomic status would correlate with higher rates of maternal anxiety. So my project consisted of 27 participants, 17 of which were male, and the average age of these participants was 3 months old, and they came into the lab with their mothers. I first distributed the still face paradigm. So this is used to mimic stressful conditions for an infant in 3 steps. So this first step here is phase 1, which is called the baseline phase. So that's where we instruct the mother and child to interact under normal conditions for 2 minutes. We then go into phase 2, which is the still face phase. So we instruct the mother to completely ignore her infant's cues, sit across from her infant, and really have a still face for 2 whole minutes. And then finally, phase 3, which is the reunion phase. So the mother and the child return to normal conditions. So the variables measured, I first measured infant affect. So that was broken down to 3 subscales. So positive, a happy baby; neutral, unengaged baby; and then negative would be a fussy or even a crying baby. And then on the mother's end of things, I coded for maternal sensitivity, which is the mother's ability to respond to her infant and interact with her infant. So the first subscale would be intrusiveness. So that's how maternally led this interaction is. Acknowledging would be how infant led the interaction was. And it should be noted that when a parent and child do interact, you want this interaction to be much more infant-led, so the infant's cues are really being tapped into. And then the last subscale would be positive affect, so that's how happy and engaged the mom is when interacting with her child. I then distributed a questionnaire to mothers, and this consisted of different aspects such as socioeconomic status, and then Edinburgh, STI, and PSS. All listed a bunch of statements, and the mother would respond to how often she felt that way since she had given birth to her child. I then took a sample, a hair cortisol sample from the mothers, and cortisol is a stress hormone that is released in the hair, and it provides a month-by-month timeline for periods in which a stressor is pertinent. Going into my results and conclusions, this first table here shows the correlation between maternal anxiety and socioeconomic status. As you can see, highlighted in yellow within the table, maternal education and hair cortisol negatively correlate with one another. So from this, we can conclude that higher levels of socioeconomic status are associated with decreased levels of anxiety based on this negative correlation. This next table here shows the correlation between maternal anxiety and maternal sensitivity. So baseline and reunion intrusiveness both positively correlate with hair cortisol levels as indicated and highlighted in the table. From this, we can conclude that mothers with higher levels of anxiety indicated by hair cortisol are likely to override or even ignore their infant's cues because, again, intrusiveness is how maternally led this interaction is. Now, this table here shows the correlation between maternal sensitivity in the baseline and the reunion phases. And now remember, during the still face phase of this paradigm, mothers are instructed to not respond to her infant's cues. So for all 3 subscales of maternal sensitivity, intrusiveness, acknowledging, and positive affect, the mother scored a 0. So when comparing the two, Intrusiveness, positive affect, and acknowledging correlate with one another in the reunion phase. And from this, we can conclude that mothers made no effort to make up for the lost connection between her and her infant during the still face phase. Instead, the mother reverted to the parenting techniques that they always exhibit. This table here shows the correlation between infant affect in the still phase and the reunion phase. So the still phase is the period of stress for the infant, and the reunion phase is the period directly after stress. So infant affect in the still phase positively correlates with infant affect in the reunion phase. So we can say that infants experience the same emotion during and immediately after a period of stress. And this reunion phase was only 2 minutes long, so it does suggest that infants at this age, 3 months old, do need longer than 2 minutes to regulate their emotions and return to expressing positive affect. So the key findings of this study, socioeconomic status impacts parent-child interactions through stress mechanisms. In this study specifically, greater maternal education, so higher, which is an indicator of higher socioeconomic status, correlates with lower levels of maternal anxiety. I would like to thank my mentor and my research teacher, Mr. Garbarino, and also Mr. Victoria. These are my references. Thank you so much for listening. Please let me know if you have any questions.
All right, thank you, Caitlyn. Is there anyone on the floor that has any questions?
Caitlyn, so I'm aware that it's thought that before birth, maternal anxiety has been linked to autism, or at least that's one theory. So I want your thoughts on that. But I was also wanting to know your thoughts on its linkage to hyperactivity and ADHD as well.
Well, in this study, we look at maternal anxiety as something that really manifests itself after this child was born and all of a sudden they're caring for a new life. So I guess it would be anxiety towards providing for their child. So looking at things such as autism and ADHD and things like that, I think what maybe you're describing is something that the anxiety that was already there, not necessarily related to providing for their infant, but in this study, we were more concerned about the anxiety that the mother feels based on not having enough income or not knowing what meal she's going to provide for her infant next, things of that nature.
Okay.
All right, thank you, peers. Anyone else have a question?
Hi, Caitlyn.
Hi, Miss Vanita. How are you?
Good, how are you? Great presentation. My question is, what was your rationale for picking a time of 2 minutes, and how do you see that sort of translating to real world situations?
So I first picked 2 minutes because, well, I knew that I wanted each phase to be the exact same amount of time. So that kind of came down to this still phase because that's when the infant was under the most stress and the conditions would be the hardest for them. And I really ideally would have wanted to do that 5 minutes. The could have lasted. A 3-month infant can't really last under those conditions for 5 minutes, crying in front of the mom and being so upset like that, and having the mom not be able to do anything. First off, the mom after a long time would probably just pick up her infant and forget about the whole paradigm. And I think it would just be pretty challenging for the infant. So at 3 months old, I thought 2 minutes was appropriate for each phase. And then again, I wanted it to be the same exact amount of time per phase. So that's why I went with 2 minutes.
All right, is there anyone else with a question? Okay, since there are no more questions on the floor, we'll move on to Oliver Rayner, another student from Mamaroneck High School.
Hey guys, I'm Oliver. All right. All right, is that working? All right, cool. All right, hello, my name is Oliver Rayner. I'm a senior at Mamaroneck High School, and my project is on an analysis of the contact angle of ionized fuel droplets on porous emitter substrates in the context of an electric propulsion system for CubeSats. So sending things into space, anything really, is wildly expensive. Let me put this into perspective for you. Sending about 1 kilogram of mass into space, about 6 apples, will run you about $35,000. And if you scale this up to most modern day satellites, communication satellites, which weigh about 6 tons, that'll run you about $190 million. Now, this number, this dollar figure, is a huge limitation to the field of space and different satellites and all that. So currently, the focus of the industry is trying to reduce this cost as much as possible. And by scaling down the weight and size of satellites through the use of something called CubeSats, we can reduce this cost as much as possible. Now, in the past, we used to use chemical propulsion to maneuver large satellites throughout around in space and all that. But now with smaller satellites, which we can use something called electric propulsion. Now, electric propulsion has the trade-off of producing less thrust compared to chemical propulsion, but also has much higher efficiencies. Seen in this table from Zhang in 2003, you can see that electric propulsion is about 30 times more efficient than chemical propulsion. This makes it perfect for CubeSats. Now, my project focused on a type of technology called the IEPS, which is a form of electric propulsion system for CubeSats. And it has a efficiency value of around 25,000 or 2,500 newtons times seconds per kilogram, making it highly efficient compared to traditional chemical and electric propulsion systems. Now, this technology uses a type of fuel called EMI-BF4, which is an ionic salt, and it works by electrospraying this at a tip called the Taylor cone, circled in red. And by doing this, it electrosprays it, and after accelerated by an electric field, creates thrust. But currently, this engine has a problem of short circuiting. And this happens when the ionic liquid, as shown in this diagram on the right, floods the emitter grid. And when this happens, it short circuits the entire engine and it goes up in a bit of a plume of smoke. And that's very bad. That causes the engine to become inoperable and the emission stops after that. So one way, this is a diagram of it happening here. This is before and after. This is actual flooding of the emitter grid. But one way of tackling this is by looking at something called the contact angle. Now, the contact angle of a substrate or with a liquid is essentially a quantification of how hydrophobic or hydrophilic a substrate is. Now, by looking at different substrates of the IEPS that produce higher contact angles, we can hypothesize that this will reduce the chances of surface wetting as much as possible and increase the performance of the IEPS. So my study looked at 5 different substrates, NG18, 38, 78, and 79, all of which were identical, and one last substrate called the AG56 substrate, which was sent to me by NASA to test on the IEPS. Now, first, we used water as a control to measure with EMI-BF4, the ionic liquid that the IEPS uses. And we were able to test 10 trials for each substrate with 1 droplet per trial. And then moving on to EMI-BF4, we were able to do the same thing. And the water served as a control for each contact angle with EMI-BF4. Now, first, to saturate each substrate and for testing, a liquid droplet was placed underneath the substrate. And this allowed the ionic liquid or the water to passively move through the substrate without getting any liquid on top of it to interfere with the contact angle. Secondly, each droplet was measured according to this diagram here on the right to find the contact angle, the width and height, and as well as using a visual software to take pictures of each droplet, we were able to take up different markups of the droplet in terms of width and height and compute a contact angle using the formula shown in the bottom left. And from that, we were able to get a margin of error with a more of a visual contact angle compared to a mathematical contact angle. Now, before I move into the results, it's important to clarify that with water, we can reuse each substrate. So when we were testing the control values with water, we were able to heat up a substrate and evaporate all the water and then perform the test again. But with EMI-BF4, ionic liquid cannot be evaporated by heating unless it's extremely high heat. So we were only able to use about 5 droplets on each substrate before having to dispose of it. Now, from these experiments, you can see that we had different contact angle values for both water and EMI-BF4. For instance, NG18 had an average contact angle value of about 78.235 degrees with water, and with EMI-BF4, about 56.724 degrees for NG18. And that's all the same with each respective substrate, and I compiled them all into these two tables right here to make it a little bit easier to read. The top one for water droplets and the bottom for EMI-BF4. And from these, AG56, circled in red on both, outperformed all the other substrates, having the highest contact angle for both water and EMI-BF4 testing. So essentially, throughout this study, the EMI-BF4 droplets were much flatter than the water droplets. And this was expected due to the lower vapor pressure of EMI-BF4. So that was a good sign that the experiment is doing what was expected. From this, AG56 outperforming almost every other substrate was not much of a surprise, as it was manufactured with higher precision tools than what we currently had available in the lab. But AG56, the substrate, it had a higher contact angle by about 31% compared to NG79, the second runner-up, and about 117% compared to other substrates, as in NG18, 38, and 78. Now, the significance of doing this work is more on fueling different technologies in the field of microsatellites and different space propulsion technologies. Through the use of CubeSats, interplanetary missions and different weather mapping abilities, communications networks, and education and research in terms of laboratories and universities all become more available. And as global warming becomes an ever more prevalent issue in today's society, using the CubeSats to track different forest fires, weather patterns, and all that kind of stuff is a very useful tool in trying to tackle and control global warming. I would like to thank my mentor, teacher, and parents for their support and guidance throughout my research career. These are my references right here. And if you have any questions, feel free to ask them now.
Anyone in the audience have any questions? Thank you, Oliver, by the way.
No worries. Thank you.
Okay, if no one else has a question, this is a really cool project. How popular are CubeSats right now?
Right now, CubeSats are on the rise in terms of popularity, as in you see a lot of smaller companies such as Spire, for instance, which is based out of Glasgow, is a very small different startup in terms of commercial launching in terms of CubeSats and sending things up into space. Right now, they're used in terms of more weather mapping, communications, and stuff like that, as in establishing very large grids around the Earth in terms of communications, which is really cool, which we haven't really seen. It's like a swarm, almost like a swarm of bees or something like that.
All right, thank you. Is there anyone else that has a question?
Hi, Oliver. My question to you is on the size of your CubeSat that you're trying to build, and and the electricity as a component is the only component you looked at.
So in terms of electric propulsion and stuff like that, there are many things that ties into electric propulsion, and there are different classifications of engines. So what makes an engine electric propulsion in itself is using electricity to create propulsion. But traditionally, you see different like field emission engines. They're called FEEP engines, and they create different electric pulses using xenon gas and all that. And my engine, the IEPS, is different than that by using an ionic liquid or a salt, EMI-BF4. So what it does is it breaks up the ionic liquid into different molecules. And from that, it's then accelerated, which creates thrust. But these are all going on different satellites, which are about the size of a small shoebox. Or, I mean, some are even the size, well, I have a Rubik's Cube right here. Some are even about this size, which is really crazy. But the engine itself is about, I guess, the size of this little rubber duck that I have. So you can stack them, you can throw them together in many different ways. It's very modular. CubeSats are very cool.
Thank you. That's very nice.
Is there anyone else with a question before we move on to our last presenter?
Yes. Hi, Oliver. I'm Matthias from Pelham. Sorry, DeBella. I'm sorry. You're next. So I was just wondering, of course, SpaceX has been bringing about a sort of revolution in cost reduction, which means that more and more satellites can be launched. And so they've been launching their Starlink satellites for a while now. And so astronomers have been complaining because these super small satellites, when they enter into orbit, they're causing disruptions in astronomical observation. So what do you think about how would you be able to minimize the impact of CubeSats and other similar small low orbit satellites on science?
On science, well, I mean, that's a really great question that honestly I don't really have a direct answer to, because I mean, you have a kind of clashing, I guess, of two different fields, right? Of one, say, by sending things up into space, we're able to get a better view of our solar system and our universe, right? But what if you want to do it from Earth, like from below the atmosphere? Then you have things clouding its view, right? So, I mean, I guess there's a little bit of an argument between the two sides there. I don't really have a side. I couldn't really say, but to be honest, I don't really have a direct answer to your question. I guess I could tell you about space debris if that's what you're wondering or something like that.
I mean, yeah, it's difficult. It's very fascinating, though. Thank you very much, Oliver. Good presentation.
Thank you, Matthias. Thank you.
All right, DeBello, did you want to say something?
Yeah, I'll ask a quick question. I saw that you noted that AG64 performed very well from both a water and what was it, BF4?
EMI BF4, yeah.
Yeah. So do you think that there's any other applications for AG64, and is it kind of like cost effective for other things that are trying to be hydrophobic and have the similar contact angle?
Well, so these substrates, the one manufactured by NASA, the AG56 substrate, they're all different cuts of porous glass silicate. And by all means, they're really cool and they're great substrates for the IPS because they're porous and they do allow things to saturate through them. And you can bring the liquid through the substrate, which is really neat. But in terms of hydrophobicity, or in terms of keeping water off of things or making things more hydrophilic to an absorbing water, that's a little bit out of my field or my scope of knowledge on this topic. I'm focused more on the space side of things, not really the chemical side of things. So I mean, there are a bunch of great labs that do work. Like the lab next to me in my building was working on a completely hydrophobic material, a spray, which they started marketing not too long ago. So that stuff is really cool, but not really in my field.
Thanks very much, man. I actually just flew out to space right now. And as you can see, we need more CubeSats out here. So make sure you get on it. Great research, man. Good job.
Thank you. Thank you.
All right. Thank you, DeBello, for that. Does anyone else have any questions? No? Okay, so let's move on to our last presenter, Peter Way from Pelham Memorial High School.
Hello. All right. Hello, my name is Peter Way, and today I'll present to you my research, which is Syntaxin-4, a potential target to combat obesity and aging. Currently, obesity has become a global pandemic, and right now, there's over 650 million adults worldwide that are currently obese. In 2016, the CDC listed aging as a major risk factor for obesity, which contributes to this large number of obese adults, who are at a much higher risk to develop other diseases, including cancer, diabetes, heart disease, and high blood pressure, which ultimately, left untreated, can become fatal. Currently, our obesity treatments, such as exercise, dieting, and surgery, have been unable to deal with the rising trend of obesity. So right now, there needs to be a new solution. So obesity occurs when white fat, which stores energy, increases, resulting in weight gain. However, humans also possess brown fat, which acts to burn off energy as heat through the protein UCP1. Therefore, many studies have suggested that targeting brown fat then can result in weight reduction. However, it is known that brown fat mass and activity deteriorates with age. So as infants, we do have a lot of brown fat. However, as we get older, we start to lose it, resulting in little to no brown fat in adults. And this trend of decreasing brown fat mass and activity has been correlated to an increased risk of metabolic diseases such as diabetes, obesity, and cardiovascular disease. But if you could prevent this process from happening, you could potentially lower the risk of obtaining these metabolic diseases, and therefore prevent us from getting them as we become older. However, we don't know how brown fat loss with aging actually occurs, which prevents us from targeting this process to combat obesity. So in my study, I was looking to determine and explore the cell death mechanism responsible for brown fat loss with aging and its therapeutic implications against obesity. And I had two goals in my study. One, determine what this mechanism is, and then two, to see if inhibiting this mechanism then would actually prevent brown fat loss with aging. And in this study, I used the UCP1 syntactin-4 knockout mouse model to study brown fat loss with aging, as this model was developed by my lab to depict human brown fat loss with aging. So in order to screen out possible mechanisms, I looked at the expression level of cell death genes and noticed that the significantly expressed cell death genes were associated with a form of cell death known as pyroptosis. I didn't look at the expression level of pyroptosis pathway genes and found them all to be significantly expressed. And then I performed another experiment to verify these results and were able to show that the knockout mice indeed had greater expression of pyroptosis genes, and then this increase was indeed statistically significant, suggesting that pyroptosis is the cell death mechanism. However, to determine whether this is true or not, I needed to measure the protein level of procaspase-1 and cleavcaspase-1. So here, having lower procaspase-1 protein level accompanied by greater cleavcaspase-1 protein level will indicate the presence of pyroptosis. So I observed that the knockout mice had lower procaspase-1 level, meanwhile the wild-type mice had higher procaspase-1 level. Meanwhile, the knockout mice, though, had greater cleavcaspase-1 level, while the wild-type mice had lower cleavcaspase-1 level, indicating that pyroptosis was indeed occurring in the knockout mouse. I then quantified both of these bands and was able to show that these differences were statistically significant, showing that pyroptosis is indeed occurring. As another measure, I also performed cleavcaspase-1 staining on wild-type and knockout brown fat tissue samples, and was able to observe greater cleavcaspase-1 staining in the knockout mice, which suggests that pyroptosis is occurring. I didn't quantify the staining, was able to verify that knockout mice indeed had statistically significantly greater cleavcaspase-1, which indicates that pyroptosis is indeed occurring. So after confirming that pyroptosis is occurring and that it is the cell death mechanism, I wanted to see then if inhibiting pyroptosis would actually prevent the process of brown fat loss with aging, which my mentor agreed to do after I suggested it to her. So when we completed the experiment, I observed that our treated knockout mice with the inhibitor VX765 actually had greater brown fat mass compared to their untreated counterparts, and that this difference was statistically significant, showing that our inhibitor was actually able to prevent brown fat loss with aging by inhibiting pyroptosis, providing further evidence that pyroptosis is the cell death mechanism that's responsible for causing brown fat loss with aging. So overall, my study is the first to demonstrate that pyroptosis is the cell death mechanism causing brown fat loss with aging, which highlights a novel function of pyroptosis. Also, we are the first to show that inhibiting pyroptosis then can prevent brown fat loss with aging, which highlights a potential novel medical application of pyroptosis inhibitors. So, as I mentioned before, our inhibitor VX765 was able to prevent pyroptosis and thereby actually prevent brown fat loss with aging. And by doing so, we could potentially decrease the risk of obtaining metabolic diseases such as obesity as we get older, and therefore prevent young adults and teens from becoming obese as they get older, thereby decreasing the number of obese adults and also the number of deaths associated with obesity. In this approach, where we try to target the aging processes that lead to obesity rather than obesity itself, we not only effectively prevent obesity, but also the other diseases that come with obesity, such as heart disease and diabetes. So to continue this research, I want to see if inhibiting pyroptosis then with our inhibitor would work on regular mice in preventing brown fat loss with aging, and determine the effects of preventing brown fat loss with aging in combating and preventing obesity. I'd like to acknowledge my mentor, Dr. Darwin Fung, who works at Albert Einstein College of Medicine, the lab principal investigator, Dr. Jeffrey Pesson, for giving me the opportunity to work in his lab, my science research director, Mr. Stephen Baltakis, and teacher, Mr. Joseph DeBello. These are my references. And that concludes my presentation, and I'm ready to take any questions.
All right, fantastic, Peter. Does anyone in the audience have any questions? What is the normal function of pyroptosis?
Sure. So as I mentioned before, that pyroptosis causing brown fat loss with aging is a novel function that my study was the first to find. So typically, pyroptosis is actually used by the immune system to stop infection. So, for example, if a bacteria invaded your body, and let's say it invaded your immune cells, so what would happen is that those bacterial invasion will actually trigger the immune cells to undergo pyroptosis in order to die and actually stop the spread of the bacteria infection into your body by just killing it off right there. So, yeah, that's the normal function of pyroptosis.
There was a question in the chat.
Yes.
If you can answer that.
Sure. Has it been shown that higher levels of brown fat lead to lower levels of white fat? So that's a very good question. So it's been shown that having high levels of brown fat, or rather, it's been shown that leaner people tend to have more brown fat, and it's been shown in multiple mice studies that having greater brown fat as you get older actually prevents you from becoming obese, especially in certain areas such as your visceral fat that could actually lead to serious health problems. So while it's not been, I would say, indefinitely proven that higher levels of brown fat lead to lower levels of white fat, it's been shown to increase health exponentially and make it a lot harder for you to become obese. So I guess sunshine energy also is another question. How might this application be used in the real world? That's a good question, right? Because we're trying to figure out a way, how would we prevent obesity, especially as you get older and become more susceptible to obesity. So the idea is that you would have to start when people still have a lot of brown fat. So typically, children have a lot of brown fat, and most likely if you were to use, let's say, my inhibitor to prevent this process, you would try to go, when the kid is an adolescent, so like 12 years old, and then just treat it long enough so that by the time they're an adult, they have enough brown fat that their chances of getting obesity is dramatically reduced, and you don't have to use that treatment. In other words, you would have a lot of brown fat if they're like 20s and 30s, and that prevents them from becoming obese. Is that it? To answer the question? I guess so.
Is there anyone else with any other questions? No. So a really big thank you to our presenters from Pelham and Mamaroneck, our viewers, and everyone asking questions, and a huge thank you to our viewers on our YouTube live stream as well. As I said in the beginning, this is the first time we've ever done something like this, so thanks for bearing with us as we iron out the kinks. We look forward to doing more events like this in the future, and now I'll go back to Mr. Veltakis and Mr. Garbarino.
Mr. Garbarino, would you like to jump in real quick as we wrap this up?
Sure. I'd like to say congratulations to everybody who presented. You did a terrific job. It was really great to hear your presentations and to see students interacting again after such a long time of being home isolated with social distancing. This is the most fun we've had working with students in a long time. So thanks for everybody showing up and doing such a great job, and staying till the end and listening to each other's presentations, asking great questions. And this was terrific. And I just want to thank everybody at Pelham for putting this together and for reaching out to us to participate. We'd love to do something like this again. This was awesome. Thanks so much.
Thank you, Mr. Garbarino. Mamaroneck, presenters, thank you. Students that stayed and watched, parents that participated and watched from both venues, we appreciate your support. We also like to, again, thank Alex Wolf for helping with the media, Steve Ravita. Thank you very much again for being out there and having this with YouTube Live. Mr. DeBello and I, special thanks again to Dr. Callahan, who has just been incredible throughout the entire process here at Pelham, helping our students along the way. Great job, everybody. We hope to see you all again in the future. Stay safe. Have a great night.
Great.
Nice job, everybody.
Have a good night, everybody.
And see you about take it.
Bye-bye.